I'm very pleased today to introduce Dr. Jason Wallach as our speaker. Jason is very local to us. After receiving his bachelor's degree in cell and molecular biology from Indiana University, Pennsylvania, he received his Ph.D. in pharmacology and toxicology at the University of Sciences here in Philadelphia, up at 43rd Street, where he's now an assistant professor in the Department of Pharmaceutical Sciences, as well as the Substance Use Disorders Institute. He holds an adjunct appointment at Cooper Medical of Rowan and is an author of multiple publications in peer-reviewed journals, as well as numerous book chapters and popular press articles. His work as a chemist is focused on drug development for several therapeutic areas, including neuropathic pain, neurodegenerative disease, depression, cancer, and his research includes investigations of structure-activity relationships, drug tolerability, target selectivity, multi-target drug design, and biased ligand development. Now, his research has attracted significant attention, as you know, because of its focus on the therapeutic potential of innovative psychedelic compounds. These may have breakthrough potential for a wide range of psychiatric disorders, including mood, anxiety, stress, PTSD, and substance use disorders. And as noted in the flyer that we've been sending out this past week, his persistence in this research area, even when funding was really tough, has paid off. His work has attracted the attention of a mental health care company, Compass Pathways, enabling him to establish the new Drug Discovery Center in his laboratories at 43rd Street. So in today's talk, he will give us a peek behind the curtain of drug design and testing in this rapidly growing field of the psychedelics. Jason, thanks so much for being with us today.
Cool. Yeah. Thank you so much for having me, and thank you, especially Anna, for organizing, putting all this together, working with me over the past few weeks to get this together. So the talk I'm going to be giving today is called Return of the Psychedelics, Structure-Activity Relationship, Studies of N-benzyl-phenethylamines at the 5-HT2A receptor. And this is like a project that has been ongoing for about 10 years. I actually started it when I was a Ph.D. student at University of the Sciences as well, and it's slowly evolved over time. I'll do some disclosures at the end. This work is completely independent, though, of any type of industry relationships that I have. And although I'm the one presenting, it wouldn't be possible without the work of many students and collaborators who I will acknowledge throughout the talk. So, let me see here if I can advance. Okay, cool. So I want to do a little bit of kind of background just so everyone is on the same page before I talk about kind of heavy-duty medicinal chemistry, SAR. And so the class of compounds I'm going to be talking about today are the classical hallucinogens. Now, initially, the term psychedelic was coined by Aldous Huxley, the author, and Humphrey Davy, a scientist, to describe the unique pharmacological effects of this class of compounds, the classical hallucinogens. In more recent years, with this resurgence of interest in these types of compounds, the term psychedelic has kind of lost that specificity, and it's now used to describe other pharmacologically distinct classes of compounds like the empathogens, for example, MDMA, as well as dissociatives like ketamine. And I have been scolded at conferences for continuing to refer to the classical hallucinogens using the term psychedelic. So I'm trying now to not offend anyone, even though, in my opinion, there's good pharmacological and historical rationale for preserving the name psychedelic for this class of compounds. Regardless, the classical hallucinogens certainly represent a core component of the psychedelic compounds. And classical hallucinogens have been used by humans for many thousands of years. There's archaeological evidence as well as evidence from art of various cultures around the world of the use of these compounds. And so there are a number of which come from nature that are important to point out. So the magic mushrooms, which contain psilocybin and psilocin. LSD is not a natural compound, but it is derived from a number of natural compounds, for example, LSA, which is found in morning glory seeds. And then, of course, we have mescaline from peyote and other cacti like the San Pedro and various Trichocereus species. And then we have ayahuasca, which has DMT as well as compounds called monoamine oxidase inhibitors to make it orally active in the tea that it's ingested in. Chemically, the kind of natural or natural analog psychedelic classical hallucinogens come really from two broad chemical classes, the indole compounds and the phenethylamines. The indoles can be broken into two classes, the sort of simple tryptamines and the lysergamides. Representative tryptamines are psilocin, the active component in magic mushrooms and the active metabolite of psilocybin, and DMT, the active component in ayahuasca. These are very structurally related to serotonin, which will be important, as you'll see in a second. And then LSD, which has this lysergamide structure here. It's a bit more complex, but it contains a tryptamine core as well. And then we have the phenethylamines represented by mescaline. And within these broad categories, there are many sort of subtypes. There are, alpha-methylphenethylamines, which can be called psychedelic amphetamines. There are N-benzyl-phenethylamines, which I'll be talking about later. So that these sort of branch into various classes, but these represent the core structures. All of these compounds are believed to exert their kind of core psychedelic effect through a single type of serotonin receptor called the 5-HT2A receptor. And I'll talk a little bit more about that. So there are 7 families of serotonin receptors currently recognized. 5-HT2A comes from the 5-HT2 family. There are 2 related receptors, 5-HT2B and 5-HT2C, and I'll talk a little bit about those later. Now, all of the families of serotonin receptor, except one, are what are referred to as GPCRs, or G protein-coupled receptors, and they're called that because when they are activated, so they sit in the cell membrane, when they're activated by, for example, serotonin, they undergo a shape change from the inactive into the active state. And that active state is able to activate G proteins inside the cell. And once these G proteins are activated, they go on to induce a whole host of what are called second messenger signaling cascades, basically complex metabolic pathways that kind of branch out from the activated receptor and this signal transducer. And you get, as you might imagine, very complex effects. These effects are cell type dependent. And they're also dynamic even within a given cell, can vary over time with repeat stimulation, with stimulation of other receptors. And so you get very complicated signaling pathways. With respect to psychedelics, we're still really working out which of these signaling pathways are important in both the therapeutic as well as the kind of psychedelic, psychoactive effects. So a little bit more about 5-HT2A. It's very abundant in the central nervous system. Not surprisingly, that's kind of why we think these compounds exert such profound effects on the central nervous system. In addition, though, they are also abundant in the periphery. So they're found in smooth and skeletal muscle, in blood on platelets, in kidney and liver. And so they're certainly not confined to the central nervous system. And although we think a lot of the core therapeutic effects are mediated by 2As in the central nervous system, there's increasing evidence that targeting peripheral 2A receptors may also have some interesting activity, and I'll show you an example of that here in a second. 2A receptors are found throughout the brain. They're especially abundant in the cortex, particularly on layer 5 pyramidal neurons in the prefrontal cortex, but throughout the cortex regions. Those are going to be postsynaptic 5-HT2 receptors. Although those have historically gotten the most attention, in reality, 2A receptors are found on many other cell types as well. So first of all, you have postsynaptic 2A receptors. You also have presynaptic 2A receptors. In addition to being present on these excitatory pyramidal neurons, you have 2A receptors on inhibitory GABAergic interneurons. And you also have 2A receptors on glial cells. And so historically, scientists have focused a lot on these layer 5 pyramidal 2A populations, but they've focused much less so on the interneurons and glial cells, as well as the pyramidal neurons in other cortical layers. So we have a lot more to do to kind of map out the physiology of the 2A receptor. But like many physiological systems in the brain, it is incredibly complex, and it's going to be difficult, and I think many decades before we really understand what the heck is going on here. So just to show you the recognition of the importance of 2A receptors outside the brain, this is some work that was done by Charles Nichols' lab. And this was a serendipitous observation. One of his, I think it was a postdoc student, just had an idea that he wanted to try from some work he'd done in his Ph.D. to look at the effect of a psychedelic compound, DOI, with respect to its ability to influence TNF-alpha. And so they've done a bunch of follow-up work, and they basically observed that DOI can sort of block the inflammatory response induced by TNF-alpha. And so this is really interesting, exciting work. They've demonstrated some relevance in a number of non-human animal models of inflammation, and that's really exciting. It does not appear that the psychedelic effects obviously have anything to do with this. And in fact, the effect is incredibly potent, at low picomolar potency to induce this TNF blocking effect, TNF-alpha blocking effect. So anyway, very interesting stuff. I suggest checking it out. So what we're looking at here is some PET imaging work done to show the distribution of 2A receptors in the human cortex. You can see the redder color means a higher density. The radioactive signal was more dense in those regions. And they're pretty abundant throughout the cortical regions, particularly occipital, temporal, and frontal lobes. And in addition, as I said before, like we really don't understand much about how psychedelic drugs exert their psychedelic effects in humans. We have done some work and mapped out that by stimulating 2A receptors on these pyramidal neurons as well as deep cortical layer neurons, we're able to elevate glutamate output onto the pyramidal neurons and from the layer 5 pyramidal neurons. So that has been kind of clearly mapped out, and there's some work to suggest that elevation in glutamate are important. These elevations in glutamate are important in the psychedelic experience that these drugs induce. And what's interesting is ketamine, which is an NMDA receptor antagonist, so very different mechanism, also appears to do a similar thing where it elevates glutamate release onto the pyramidal neurons and results in higher output of these layer 5 pyramidal neurons. So there's some convergence. The problem, though, is that glutamate is the major excitatory neurotransmitter in the central nervous system, right? And many different psychoactive drugs are going to impact glutamate signaling. And the extent to which this is truly a unique signal, I think, remains to be determined. There's also a lot of interest in this because it may underlie, and I imagine some of you have seen this, the kind of synaptogenic changes that have been seen with psychedelic drugs, including ketamine and 2A agonists. I'm not going to go deep into this, but I will say, a problem with a lot of that literature is that most psychoactive drugs that we've looked at will cause synaptogenesis and neuritogenesis. So although those effects are very interesting from a physiological standpoint, they're not specific to any given pharmacological mechanism. You can look in the literature, for example, cocaine causes very similar synaptic restructuring as psilocybin and ketamine, and yet it's not a rapid-acting antidepressant. And so I think there's a lot of issues in the literature of people just kind of observing general physiological effects and trying to weave those effects into this narrative to explain the therapeutic actions of these drugs. So it's just something I'm quite skeptical of, and I'm not going to go too much further into it, but I want to mention that. Okay. Very recently, the structure of the serotonin 2A receptor with two different agonists bound were solved. This work was done in Brian Roth's lab, UNC Chapel Hill. So they crystallized LSD bound to the 5-HT2A receptor and got an X-ray diffraction structure solved. And then they also were able to get a cryo-EM structure with another 2A agonist, 25CN NBOH bound. The really cool thing here, though, is this protein, because they did cryo, they were able to co-complex it with the G protein, heterotrimeric protein. And this has the effect of locking the receptor into an active state or active-like state. And from that active-inactive state, you can learn a lot about how, when a molecule binds, it changes the shape of the protein to induce an active state. And we can use this information in drug design to design better, more potent drugs, as well as more pharmacologically interesting drugs. In addition to kind of, this paper goes into this, but seeing the restructuring of the alpha helical domains that make up these GPCR structures, you see shifts, 7 angstroms in TM6 moving outward when the receptor is activated. You also get, atomic level specificity where you can see the redistribution of various amino acid side chains and actually see at the molecular level, how is it that binding of a drug like LSD reorients the amino acid side chains to shift the receptor from an inactive state into an active state. And so that is really cool, exciting stuff. And this paper, will be cited, I'm sure, by many for years to come as a source of very useful information in the development of the next generation of psychedelic compounds. So there's a couple other things I want to get into before I discuss my work to just kind of highlight the complexity in understanding the pharmacological action of these drugs. And the first is something called bias signaling or ligand-directed trafficking. And this is the property that when a molecule binds to a G protein coupled receptor, I already told you that when the receptor is activated, it will signal through the G protein pathway. But G protein coupled receptors can also couple to other proteins on the intracellular side of the cell. One example is beta-arrestin. And what makes this really interesting is most drugs, most psychedelic drugs, for example, activate both pathways about the same extent. They don't show any real difference. But it is possible, we know from other receptors, that we can find and develop drugs that show selectivity for one of the pathways over another. And that's called functional selectivity or ligand bias. And actually, the first intentionally designed biased ligand was recently FDA approved, and that's a drug called olaceridine. That's a G protein biased agonist. It's biased for the G protein pathway at the mu opioid receptor. So as shown here, it's able to, by binding to this receptor, activate selectively the G protein pathway with some selectivity to not activate the beta-arrestin pathway. And the idea here is that the G protein pathway, GI, in this case, mediates the analgesic effects as well as the euphoria. But beta-arrestin is more implicated in things like respiratory depression, tolerance, and constipation. So the idea is you get less side effects with these biased compounds than you would with a unbiased compound like morphine, for example. The extent to which this will work in humans is unclear. A lot of this work was done in animal models, non-human animal models, and we don't yet really know in humans whether this is going to translate. Even though the drug was approved, the trials weren't designed to test this hypothesis, right? They're just designed to show efficacy and safety. The other thing is there are other hypotheses put forth to explain the improved tolerability. For example, partial agonism at the G protein pathway. So I'm not going to go into that, but I just want to kind of point that out for those interested in it. The bias work is fascinating, but it's very difficult to make sense of, and there's a lot of debate ongoing in the field. Okay, another layer of complexity is that G protein coupled receptors can form dimers and potentially other higher order oligomeric structures. So for example, you can have homodimers where two 5-HT2A receptors dimerize and they signal together as a unit. You can also have heterodimers where you have two different GPCRs like serotonin 2A and dopamine D2 receptor, or serotonin 2A and the cannabinoid CV1 receptor. And so you get unique signaling properties from these dimer complexes than you would from the individual receptors on their own. To make matters even more complicated, these dimers can show ligand bias that's unique from the bias observed at the monomer GPCR. So just to kind of illustrate the complexity of everything going on here. And it is very difficult to study these actually because the effect is very fleeting and transient in nature. Okay, so although I'm I'm kind of telling you and asking you to just trust me that it's the 5-HT2A receptor that really mediates the core effects of these classical hallucinogen compounds. Now, that's not to say that other receptors aren't important, and without question, the differences in the nuance between some of the compounds almost certainly involves polypharmacology, that is, interactions with receptors other than 5-HT2A. I'm not going to go deep into trying to explain the evidence to support the 2A hypothesis, but I do want to say a few things about that. And so first is that every known compound that is classical hallucinogen-like in humans is an agonist at and binds to 5-HT2A receptors. Okay? A problem is they also bind often to serotonin 2B and 2C receptors. But if we look at the relationship between the binding affinity and the potency in humans, the correlation is best with the 2A receptor. In addition, there are selective 2C and 2B agonists that do not cause psychedelic effects. So all these 2A compounds, with a very small number of exceptions that we can explain, will cause psychedelic effects, whereas 2B and 2C agonists will not. The other bit of evidence comes from the fact that we can use antagonists in humans and non-human animal models to block the activity of these compounds. So if you give a 2A antagonist like ketanserin or risperidone, which ketanserin has some, tenfold selectivity for 2A over the other subtypes, you're able to block the psychedelic effects mediated by the 2A receptor. Okay, and in the animal models, we've done genetic knockouts and various antagonist studies that show the core behavioral effects in mice and rats is definitely 2A mediated. But again, it's not to say that these other receptors don't contribute. So, in addition to the fact that many existing psychedelics act at not only 2A, but 2B and 2C, a number also act at 1A. And the interesting thing here is it's mostly the tryptamine compounds and the lysergamides that act at 1A. Most of the phenethylamines don't interact with 1A really at all, certainly at any relevant potency. And there's been a lot of work over the years to suggest that interaction with 1A might alter the effects of a compound at the 2A receptor. And so there's some interesting polypharmacology going on here that we're really still actively investigating. One of the most interesting examples, in my opinion, is a compound called 5-MeO-DMT. This is DMT with a methoxy group in the 5 position. DMT, both of these compounds are considered psychedelics and classical hallucinogens, but they are very they are pharmacologically distinct in their experience. Particularly, 5-MeO-DMT is said by most people to lack visual hallucinations. DMT and LSD are highly visual. People report very intense visual experiences, whereas 5-MeO-DMT, people say it's psychedelic, but there's no visuals, or if there are, they see like a little bit of patterns, but nothing like people experience with DMT. The other thing people will say is it's much more tactile. In that way, it feels more like ketamine, where people feel like they're expanding and shrinking and floating. And that's distinct from compounds like DMT and LSD. So there's something unique about that compound. And one of the sort of most obvious candidates to explain this is its potent activity relative to these other compounds at the 5-HT1A receptor. And so there's another compound that's interesting that I think there's some polypharmacology going on with 1A, and that's an LSD analog called LSM-775. So this is some work that we did a few years ago. This compound was brought to our attention by a group that were developing LSD-type compounds to be sold actually as legal highs. And they had said that when they tried this one out, that although it was certainly psychedelic, it just was it never reached the same level of intensity that LSD reached, and it just seemed to be missing something. And although that wasn't good if you want to make a legal high, right, to us it was very interesting because it is a anytime you see a difference, it's a question of, well, what can we learn from this difference, right? And that's one reason I love studying these legal highs, because there is human data, even though it's anecdotal. And so we characterized this compound's binding and functional activity, and we saw that, it was a 2A full agonist, comparable to LSD, about comparable potency as LSD. But the one distinction relative to LSD with its binding profile was that it was much more potent at the 1A receptor. And it was also a full agonist there, but the notable thing was it was much more potent than LSD. The other thing we were curious in, and I'll talk more about this model shortly, is the head twitch response. This is a model in mice we use to predict whether a compound is going to be psychedelic in humans or not. And it has very good predictive validity with predicting that. And what we found was the mice, like humans, were basically telling us this compound's not very psychedelic. They weren't exhibiting a strong head twitch response when just given LSM-775. And our suspicion was this was because of the high 1A activation. It was countering the activity of 2A. And so when we blocked the 1A receptor with a 1A antagonist, WAY-100-635, you actually saw then a robust head twitch response in these mice. And the head twitch is well known to be mediated by 2A. So it seems as if in both humans and mice, 1A activity is somehow able to sort of counter the 2A activation. And there's some molecular logic to this that I'm not going to go into about the competing G protein pathways, but a bit of that has been mapped out. There's also some clinical evidence in humans for this effect. Rick Strassman in the early 90s did a similar experiment in humans where he gave them DMT and then blocked the 1A receptor and saw that when you block the 1A receptor, the intensity of the DMT experience actually increased dramatically. So DMT doesn't have super high 1A activity, but it has some, and by blocking that, you're actually able to get an even more intense experience. So I think it would be very interesting in humans to block 1A and look at the effect on 5-MeO-DMT and LSM-775 to see if what we see in animals can be recapitulated in humans and how translatable these results are. And so that'll be really interesting to see. Okay, so what I want to do now is kind of talk more about some structure-activity relationship work that was done in my lab. I'll warn you, this is kind of hardcore med chem, but even if you don't quite follow all the nuance, maybe you'll get the kind of music of what I'm talking about. And so this work starts with a compound called 2CN or 25N. I was interested in this about over a decade ago because there were reports in the literature that it exhibited ligand bias. Interestingly, we found that in our newer assays that we could not observe any type of bias. But what I found interesting about it is, although 2CN is not very potent itself, when we made the N-benzyl analog, 2-methoxy-N-benzyl analog of it, it became one of the most potent of all the N-benzyl compounds. So you can kind of take any phenethylamine you want, like 2CN or 2,5-N shown here, and attach this methoxy-benzyl group to it, and the potency will increase about 100-fold. But what was most interesting to me is that you can take one of the least potent phenethylamines and turn it into one of the most potent N-bombs, suggesting there's some difference in the SAR that's happening there. The other thing we really liked was that when we looked at these 25N and benzyl compounds, we saw a reduction in the activation of the 5-HT2B receptor. So phenethylamine compounds like 2CN and almost all psychedelics, as I said before, will activate 2A, 2B, and 2C. 2C is not really a problem. It may contribute to the activity, but it's not an issue. 2B activation is a problem. 2B in drug development is considered something to be avoided at all costs. And the reason is if you activate 2B, you have an increased risk of cardiac toxicity. These drugs cause valvulopathy, and that's well understood. And when a company's developing a new drug, they make sure it doesn't bind to or activate 2B. And I think if we're going to develop these psychedelic drugs as clinical agents, 2B activity, if you're giving the drug once every few months, is by all indications, fine. But if you're going to be using any of these drugs on a regular basis more frequently, for sure you want to avoid 2B. And the FDA is almost certainly going to require it's either avoided or extensive toxicology work to show that for whatever reason, in this case, it's not as big of an issue as it is historically. So what we really liked, though, with these N-benzyls was, again, we were seeing they were less potent at activating the 2B receptor than their phenethylamine counterpart, 2CN or 25N, right? And so there was something kind of unique going on there with respect to that pharmacology. And this was in a Brett GQ assay for those interested. This synthesis, I'm just going to skip over this. So yeah, once we kind of make the compounds, you know, we do receptor binding work. The binding work I'm presenting today was done by the National Institute of Mental Health Psychoactive Drug Screening Program. The functional work, the activation of the receptor, was done by my collaborator, Dr. John McCorvey at Medical College of Wisconsin. And he ran a number of different assays on these compounds. Calcium flux assay in a flipper station at 2A, 2B, and 2C, and then Brett assays for GQ and beta-arrestin recruitment, so GQ dissociation, beta-arrestin recruitment at 2A, 2B, and 2C. I'm only going to kind of focus on a subset of this data today, but just to kind of put that out there, we're looking at activity in a number of these different assays. So this loss of 2B activity we observed in the Brett data, and to get some orthogonal evidence that this was relevant and not just like an artifact of this one assay, we also ran it in the calcium flux assay, and we saw actually in that assay virtually no activation of 2B. So this gave us, you know, orthogonal lines of evidence that the lack of 2B agonism is likely physiologically relevant. Of course, we're going to ultimately want to test that in an in vivo model to make sure, or some type of physiologically relevant model to make sure it is translatable, these assays, right? That's always important. Okay. So now for kind of the SAR bit. So kind of to set the stage for this, I still remember, you know, where this idea originated from. I was talking with a collaborator of mine, Adam Halberstadt, who does all the head twitch animal behavioral work that I'm presenting. And we were interested, he was writing his R01 application, and I was a collaborator on it, and we were trying to figure out a way to test the hypothesis that the 100-fold increase in the affinity seen when you add this N-benzyl group was mediated by this hypothesis at the time that these were able to hydrogen bond with an amino acid in the receptor. And we were interested in testing that hypothesis. But we noticed when we looked at a subset of the compounds we had that there was an alternative hypothesis, and that was that it was the substituent's electron donating or withdrawing capabilities on the ring that actually explained the difference in the binding affinity. And so we looked at a small subset of compounds we had, and we saw that trend. And then we looked in the literature, and with a small subset of compounds, we saw that trend. It certainly wasn't enough to be convincing to anyone, but it made us think we could test this hypothesis. And so to do that, we designed a series of compounds that had substituents in the 2 and 3 position of this benzyl ring called 2' and 3'. And the idea was that the substituents in these positions alter the electron density at a distant site of the ring, this 5' region. I'm not going to go into our rationale for choosing that region, but we had rationale from this sort of pilot observation that was the key site that which would be predictive in this structure-activity relationship work. So the first thing we did was we designed a series of compounds that would differentially affect the electron density at this 5' position. And the way that you can know what the effect of a substituent is something called the Hammett sigma constant. These are literature values you can look up, but they're derived experimentally on various aromatic compounds like benzoic acid, benzylamine. You can look at the effect of the substituent on things like reaction rate, equilibrium constants, and these Hammett constants have shown good utility in predicting chemical reactivity of molecules as well as pharmacology. So what's shown here in the color coding, and this is a little bit arbitrary, but red means strongly electron donating, so the substituent donates electrons.
Yeah, I got it now.
It position. Blue means a neutral effect.
Thanks
It's not
for clarifying.
donating or withdrawing. And green means strongly withdrawing. So green is pulling electron density away. And so we designed a whole series of compounds to exhibit a range of electrostatic effects on that position. Okay. So the first thing we did was to look at the relationship between the Hammett sigma value and the receptor binding affinity. And you can see that compounds the red compounds, which are highly electron donating to the C5 prime position, had the highest binding affinity, a higher pKi value. Whereas compounds in green, which had an electron withdrawing effect, right, a more positive sigma value, had the lowest affinity. In fact, these compounds, the addition of this benzyl group is detrimental to the binding. They have weaker affinity than the 2CN starting material compound. So that's an undesirable interaction. Whereas these interactions are desirable in the sense that they increase the affinity over 2CN. Now this is great, right? We were really excited to see this because it suggested there was this clear, clean electrostatic relationship. But you know, at the end of the day, correlation is not causation. And first of all, we wanted to test this a few other ways. But the other thing was, you know, if you really understand something and you've built a nice model, your model should predict something about reality. You should be able to use that model to build something. So we should be able to use this to design a compound and predict its affinity. And so to do that, I busted out the literature and went through all these Hammett tables and found two substituents, a 2 and a 3 substituent, that should theoretically have the highest binding affinity because it's the most electron donating to that position. And sure enough, we synthesized that compound, 25NB2OH3Me, and tested it, and that compound had the highest binding affinity of the entire series. So this was really cool because we can now use, you know, show that we've used this model to actually predict things that can come true, right? But at the other end of the spectrum, I was a little disappointed because we had nowhere else to go. We've sort of mapped out the full range of what's possible with respect to modulating the electron density of this scaffold. There's really nothing else, no other substituents we can put on here to increase the affinity further. So we have other ideas on how to achieve that, but this is kind of a dead end in the sense we can tune the binding affinity within this range, but we can't go higher or really lower if we wanted to, just because we've fully mapped that region of chemical space. All right, so the next step was to see, you know, what other sort of ways can we show that this is a real effect. So the first that I'm going to be showing is we looked at the relationship between the Hammett values and the potency in the calcium flux assay. So this is different from binding affinity. It's potency in a functional assay. And we saw the exact same correlation. Interestingly, compound 18 was not as potent in that assay, but regardless, the linear trend was there with compound with highly donating substituents having the most potent activity, the highest PEC50. We did the same thing in the Brett assays, and again, it's not shown here, but you see the same exact correlation. So multiple distinct measures of potency, binding, and multiple different functional assays all show this same correlation. And I said that I like the Hammett values because they're experimentally derived, but the reality is they're derived on unrelated compounds, right? Benzoic acid, benzylamines, which are similar but different. And I really wanted to have a measure that was specific to the compounds in question. And I'm kind of scratching my head and thinking about how the heck could we actually measure the electron density at the C5 prime carbon. I realized that we could use NMR chemical shifts as a physical measurement on these compounds to estimate the electron density. And so the idea here is that carbon nuclei that are shielded will appear upfield on the NMR shift scale, right? and they're shielded because they have higher electron density. So compounds in red have a lower chemical shift value. That is, they're upfield because they're shielded from the electron density. Those had the highest binding affinity, whereas those that are deshielded, more downfield because they have relatively less electron density, have lower binding affinity. And so this was, I think, some of the coolest work that we were able to do because, again, it was a physical measurement on the compounds in question. It was not literature-derived Hammett values. This was a real measurement on the compounds in question, and you saw the exact same correlation. Okay. So the kind of next bit I'm going to talk about is the in vivo data, right? So all this is great, but it's all in a test tube. And the question is, are these compounds active in animal models? And does their potency in animals correlate with the potency we're seeing in the test tube? If it doesn't, it's great, but how useful then is what we're doing? So as I mentioned earlier, the assay we used is the mouse head twitch response. So this has been used for several decades. It's when you give a mouse a psychedelic 2A agonist, they do this horizontal head twitch. And it's really fast. It's almost hard to see it. People used to film the mice and slow down the video and physically count the head twitches. Adam invented a technique where you implant a magnet in the head of the mice and you wrap copper coils around their cage. And when they twitch their head, it generates an eddy current in the copper coils, which sends a voltage to a detector. And you can actually measure the head twitch that way. And so that's much more high throughput. So this work was published that we did with Adam. He led up this work a few years ago where we showed that the potency of a compound to induce the head twitch response correlated very well with the potency of the compounds to induce hallucinogenic effects in humans. And the nice thing about this is we picked hallucinogens from every structural class available. Phenethylamines, amphetamines, N-benzyl-phenethylamines, lysergamides, tryptamines, alpha-methyltryptamine. So pretty much any representative scaffold we could, we picked. And to see such good correlation across 6, 7 different scaffolds was actually surprising. And this shows that this assay has good predictive validity. It can tell you, it can predict within really good accuracy the potency of a compound to be psychedelic in humans based on the mouse data. And in the paper, we also compared it to drug discrimination in rat, another common assay, behavioral assay used, but at much more low throughput, and showed excellent correlation with that as well. So head twitch is really reliable. There are some caveats that I won't get into, but if you use it correctly, it's a highly predictive assay, good predictive validity. It also has very good construct validity because it's mediated by 2A activation. Cool. So a couple other things about head twitch. When you're doing head twitch, it's important to do a full dose response. The reason is the head twitch response is biphasic, so you'll give increasing doses. The mice will twitch their head more times in this 30-minute window up until a maximum point where it levels out, and then it begins to drop off again. And actually, if you continue to give high doses, it will go back down to basically baseline. Some labs will only run a single dose of head twitch, and they'll say that we developed a non-psychedelic 2A agonist, and they'll say that they've shown that by showing an absence of head twitch at a single dose. Well, the problem is if you're on the low part of the ascending arm or the high part of the low part of the descending arm, you're not going to see a head twitch, even though at another dose the compound would cause a robust head twitch response. So we always actually determine the ED50, which is the half maximal dose, the dose which causes a half maximal head twitch response for that compound, all right, which would be something like here for this compound. All right, and so again, what we were looking at here was to see if the receptor binding affinity, in this case, correlated with the potency, the ED50, to cause head twitch response in mice. That should be ED50. And again, we saw a nice correlation, which wasn't surprising, but it was nice to see that compounds with higher binding affinity caused a more potent head twitch response. And also you can see this red, blue, green trend here. Compounds that were electron donating and had higher affinity were more potent in the head twitch response. And actually, some of the other green compounds we couldn't test high enough doses because we ran into solubility issues. So, we would have liked to test some of the other weaker compounds, but we just weren't able to dissolve them up to test a high enough dose to get the ED50.
Jason, I just had a question from the chat that you might want to take it before going on. It's just a technical question. How long after drug administration do you start measuring the head twitch response, and is it an IP injection?
So you can do IP or sub-Q. These were done sub-Q. With IP, some compounds will still have a bit of a first pass effect, and the N-benzyl compounds are heavily metabolized by hepatic enzymes. They're actually not active orally in humans. So those compounds we gave sub-Q. Many times IP is actually used, though, for other compounds. I believe in this case it's measured almost immediately after. I would have to just double check, because it's a 30-minute window. It may be 10 minutes, though, now that I'm thinking about it, before. But certainly that's standardized, and then you collect over a 30-minute window. There's different labs use different windows. Adams found 30 minutes to kind of capture the widest range of compounds, but it gets tricky because some compounds like DMT have a really short duration in head twitch, and in those cases you might use a 10-minute window. But there's ways to normalize the data, counts per minute and whatnot, depending on what you want to do.
Thanks.
So anyway, hopefully that answers that. And if you want to email me or Adam or look at one of the papers, I can give you the exact time. I do think it's 10 minutes, not immediately, but I'm not 100% on that. Okay. Most recently, we've begun doing some in silico modeling, computer modeling, to try to get a deeper sense of kind of what's going on with the electrostatics of these compounds, and figure out how they're binding to the receptor and how they're interacting when bound. I won't go too deep into this because I'm running low on time here, but we ran some partial charge calculations and molecular electrostatic potential surfaces on these N-benzyl compounds and looked at the Hirschfeld charge at the C5 prime carbon. And again, this was just a measure of the electron density at that part of the ring. And again, we saw very good correlation with the Hirschfeld charge calculation and the receptor binding affinity, with higher donating compounds having the highest electron density in the predicted model.
Anything? What the hell? No. The fuck?
So that's neat, and we're sort of actively exploring other modeling to look at this. And just to be clear, we're not saying that the C5, the density at C5 necessarily is itself essential. C5 could be predictive of electron density, for example, in the center of the ring as well. That's harder to model with things like Hammett and NMR shifts. Really, you can't easily, in fact. But with these in silico approaches, you can begin to ask questions like that. So the charges on any of the other carbons do not correlate. After we ran this, we looked at all the other carbons as well to avoid p-hacking, right? But once we saw this correlation, we checked, and none of the other carbons show any correlation. But that doesn't mean other parts of the ring don't. And it also doesn't mean some of the hydrogen don't, which is the H5 prime, in fact, does, as you would expect. So it gets kind of complicated. We've begun doing induced fit docking to see how the molecules bind at the receptor. And we see, sure enough, based on the work that Ryan Roth did with 25-CN-MBOH, that these benzyl rings interact with several aromatic residues inside the 2A binding site, particularly phenylalanine 339, but also phenylalanine 340 and tryptophan 336. When we started out, we were really hoping there would just be one aromatic interaction between the benzyl ring and phenylalanine 339. But when Brian Roth published this data, we quickly realized that is not going to be the case. And our hope of seeing a simple geometry to explain our SAR was out the window. And now we had 4 aromatic residues in this sort of dynamic multi-body problem that involves quantum mechanics and gets insanely complicated and difficult to model. So we've started doing some molecular dynamics simulations to try to make sense of what might be going on, but a lot of this modeling, the software doesn't capture aromatic kind of pi-pi stacking van der Waals interactions very well. It kind of underestimates the enthalpy of the interaction, and so we're kind of looking into new approaches to better model those van der Waals electrostatic interactions between these aromatic residues. But you can do really cool stuff with molecular dynamics and look at the interaction between your ligand and these various amino acids over time, for example. So this is time. Every nanosecond is a single frame, and on the y-axis you have all these different amino acid residues, and the color is just it's a heat map of the energy of the interaction. And you can see, the benzyl ring interacts strongly with tryptophan 336 and phenylalanine 339 throughout the entire simulation. And you can begin to see things like that, like, 340 is more of a transient interaction, and that kind of flips. And anyway, so you can do really cool stuff that we've begun to do. So real quick, I just want to acknowledge my lab group. I got an excellent group, really happy with them. And so that, work wouldn't be possible without them. My collaborators, Adam Halberstadt, John McCorvey, Randy Zohar, and Andrew Heim, his postdoc. John and Adam have NIH grants that funded aspects of this project. I'm a consultant and have contracts with Compass Pathways and Bex and Biomedical, although this work was independent of that. I've also received funding from the University of Sciences and acknowledged the National Institute of Mental Health Psychoactive Drug Screening Program. My former advisor, Dr. Adeboye Adejari, who let me start this work during my Ph.D. in his lab, even though it had absolutely nothing to do with what I was supposed to be doing, which I did do as well, but I'm very grateful for him for allowing me to do that. Okay.
Thank you so much, Jason. And I know that we have medicinal chemists in the audience who will perhaps ask you really specific questions, so I'll let them go first. But I think that for the people who are not straight up medicinal chemists, they'll be very interested in thinking with you about what are the next steps. You're trying some new scaffolds that are not just straight up variations on the usual psilocybin structure. So where are things in the path of development? Is it, 25% of the way there to try something in animals? And I know that the path from animals to humans is never straight. So what are your thoughts in terms of, I guess, time horizon for development of some of these things?
Yeah, that's a great question. So there's a bit more to this whole narrative. So I kind of left out a bit that we're planning to publish very soon, and for that reason, it was a little careful what to disclose. And we have patents on some of those compounds, and they do show some interesting pharmacology, including in vivo effects, but it's still very early stage. A lot of the animal models, as I'm sure you're all aware, in CNS discovery are very limited with respect to predictive validity in humans. And so that's, I think, something we're running up against. I kind of feel like you need to have a really good pharmacological hypothesis rather than kind of rely on phenotypic results in these animal models, which have historically shown a complete lack of an ability to predict whether a drug will do anything in humans or not. So, yeah, I think, we're kind of at different stages with a number of things, but, things are certainly progressing.
Oh, absolutely. I mean,
and I think there's a clear path to the clinic, which I think is nice, and a nice opportunity to have access to.
And related to the modeling and actually picking an animal model, I know obviously the head twitch response has worked really well for predicting things that will have hallucinogenic properties. What about the ongoing discussion about whether or not the hallucinogenic part is actually important for the potential therapeutic benefits or not, right? And so in terms of basically the therapeutics for these compounds, I see that as part of an ongoing discussion. I'm not in the middle of that field, but I see it and hear it at conferences. And of course, it's of interest because using the compounds with heavy-duty hallucinogenic properties requires a lot of care and a lot of monitoring and sort of hand-holding. You can't just sort of use them in the same way that you would with a simple straight-up prescribed medication with less dramatic psychological immediate effects. So what are your thoughts about that? Because I know you're using something that obviously head twitch is your index for a lot of this, which is related to the hallucinogenic piece, right?
Yep, absolutely. So that's a great question. As you pointed out correctly, there's a lot of ongoing debate in the field whether psychedelic effects are necessary for the clinical response. My suspicion, just my opinion, is it's going to depend, obviously, on the clinical effect in question. So, for example, the anti-inflammatory effects that Chuck Nichols sees, I doubt the psychedelic effects are necessary whatsoever. The activity in cluster headaches, which, a single dose of psilocybin can cause remission of cluster headaches for 6 months to a year. There's no evidence there that the psychedelic effects have anything to do with it. On the other hand, when you talk to people with depression or PTSD or substance use disorders, they can't help but describe the experience as instrumental in changing their perspective and outlook and giving them a new kind of lease on life. And although, it's hard to say that there's not some physiological change that's happening, I kind of suspect we will not have much success with non-psychedelic 2A agonists, which are easy to develop. I know there's a lot of Nature papers coming out. We've had compounds like that, 10 years ago.
Right.
They're just partial agonists. And so I don't know, but my hunch is they're not going to be so effective in some of the kind of core psychiatric indications. And this issue of, people have tried with opioids to get rid of the euphoria, right, for over a century without any luck. I think people have tried this with dissociative drugs as well, trying to make NMDA receptor antagonists that are non-dissociative. Well, every single one of them has failed in the clinic. There's probably 10 that have been tried. The only NMDA receptor antagonist that works in depression is ketamine, that's been demonstrated in phase 3 trials, is ketamine, and it's absolutely being used at fully dissociative doses.
Right, yeah.
That's not to say that dissociation is important experientially. I don't know, but I think we should be cautious of that approach.
Yeah, it's an interesting area. I mean, I think this will be one of the ongoing questions. So I'll shut up and let other people ask a couple questions because I'm sure there are some. Other folks? Questions for Jason?
Hi, this is Tom Grimm. So I'm a chemist by background. I don't know how many others there are in the audience, but I really appreciated the work that you did to indicate that the electrostatic component of that aromatic ring was important. One of the questions I had was, I think there are a couple more recent techniques now that are out for doing large-scale computational screens for electrostatic similarity. And I guess given the fact that you now have something that's like a fairly accurate X-ray crystal structure, do you think it's worth trying to scaffold hop to find something?
Yeah, and I'm very much interested in that. Now, I didn't present it, but we have tried a number of other aromatic rings to try and get at this. In all cases, we've tried so far reduced potency, but I haven't yet tried, to use some type of in silico screening or selection to make those predictions for us. It's just been, let's buy every aromatic aldehyde we can and make them kind of thing. So yeah, I really would like to actually try to predict, and I think we have the tools now to more accurately do that. That's definitely next steps, as well as look at non-aromatic things that might show favorable interactions with those aromatic rings. There are a number of cations, for example.
Thanks for the talk.
Other questions from chemists or non-chemists? It's wonderful having you right down the street or right up the road, whichever way. And, as things move along, Jason, we're obviously going to be watching out. And if something comes up that you think is exciting and you want to talk with us about, we will be very welcoming. So just keep us in the loop.
Yeah, thank you so much for having me. Thank you all for attending.
Thank you very much. Thanks everybody for logging on.